So the next thing I'm going to go into is the genes, or the genetics of this. But I hope you've had a little bit of time to think about this question, and we'll take a couple of minutes just to. What do you think is going on in the family at the phenotypic level, from what I've said so far? Any thoughts? Okay, who thinks that? Who thinks they have language impairment? Put your hand up. Sorry.
Yeah.
Sorry?
Should we say something?
Yeah.
Okay.
Sure, go.
Just because of they show that they have grammatical problems showing the right picture. Yeah, this is it.
Does everyone agree with that? Anyone want to counter it? Okay. So, yeah. Yeah, they do. But I mean, it could be. So some people argue that these problems are actually much more subtle than their problems with speech development. So one argument would be that actually, if you grow up with a primary problem with being able to fluently speak, you have sort of impoverished language as a consequence, and that this messes with your. I mean, does anyone have any feelings about that? Whether they're. I have to tell you, there's no wrong or right answers here. This is just something that everyone's kind of arguing about, and I don't know what the. I mean, I have a position on it, but I don't know what the. Any thoughts? You don't care? Do you think it's an important question to decide whether, because it could, so an argument that we heard a lot when we were first talking about this was, yeah, they have a general motor problem which happens to impact on speech, and then it's not going to tell you anything interesting about language.
I think it is important to ask the question, as you said at the onset, there is no gene for language,
yeah.
But it is important to ask, is this a gene for language?
Yeah.
That's one of many genes that is for language. I think that's a reasonable question to ask.
I mean, I would say it's kind of an ill-posed question because language is not a monolithic thing. So therefore, I think maybe it makes more sense to say, does it affect a certain aspect that's crucial for language?
Yeah. Bruno.
But you said it was mostly a motor problem, if I understood, motor problem. But if they also have the problem, as you said, with the shoe and the pencil, then it's larger, and then maybe it covers actually.
So
more.
what with?
The
with the yes.
Yeah. So then it's not just about articulatory muscles and stuff. So.
Well, what would you like to know? If I'll come to you in a second, but what would you like to know before you say this is a general motor disorder? I mean, some people have said this is just a general motor disorder.
Well, you could test their hand motricity. So.
Right. They don't have limb dyspraxia. Now, so it seems like they have a particular problem with orofacial sequencing. There has been a paper that claims that they're not very good at repeating rhythms when they bang, but I think those effects are very subtle. I'm not sure if I believe them so much, but I would like to see more on that. But so people have argued that either. Welcome back. We're talking about kind of specificity of the K family's problems. So some people would argue. Sorry, some people would argue that. Yeah, it wasn't on. I think. No. Yeah. Some people. That's better. Some people would. Yeah, it wasn't on, which is why I was so quiet. Some people would argue that the sequencing that we do during speech is one of the most complex motor acts that we do, and maybe they would have a problem if you gave them a sufficiently demanding motor skill task. This issue will come back a bit later, so think about this.
In their play, do they play basketball or football?
They're not. I don't know how sporty they are, but I know that none of them are particularly musical, so it's a bit difficult to know whether, if one of them had learnt a musical instrument, we could. I thought it might be good to look at what they do on kind of PlayStations and things like that. So anyway, that's another dimension of knowing how specific the motoric problem, whether it's more about their orofacial learning than others. Can I get to Tineka's question?
So even if it's not completely specific to that only language is affected, but also some other domains are affected, it can still informative about language processing.
Yes, and that's really my position, is that if we want a really pure dissociation, if we're only going to believe stuff when it's purely, people get very excited about specific language impairment. Some people describe the K family as suffering from specific language impairment. From a technical perspective, they don't. And the reason is because you can't give somebody a diagnosis of specific language impairment unless they have an IQ, a verbal IQ that's at a certain level. And also, if they have speech apraxia, that excludes them from getting a diagnosis of specific language impairment. Now that tells you something about how we have to be a bit careful about our diagnosis. And I think in a way what Nina was showing, which was pretty much that every person with aphasia is sort of different, right? Well, you kind of want to dimensionalize it and understand the different things that are going on, and it's interesting to know that. But you wouldn't go, oh, this person's just not interesting at all because, you know, throwing them out because the profile is uneven. And I also should say that there are kids with, it's gone off again, I think. Yeah. So I think that what we need to know is understand the dimensions of the phenotypes that we study, understand the genes, which I'm going to show you now how we've found the gene for this, and then just build our theories around it. But if we can't decide on, thank you, we can't decide on what's important and what isn't based on this. I also think that it's worth knowing what we know about genes as well. Most of the disorders that we study do hit multiple cognitive phenotypes. But what this isn't is this is not a typical intellectual disability syndrome. So we study a lot of intellectual disability syndromes in which there are speech and language. Sometimes they're completely non-verbal, these kids. Sometimes they have very severe speech and language problems. This is not a classical intellectual disability syndrome. This is something different. And I would say it's primarily childhood apraxia of speech, actually. But
I just wanted to add that amongst the myriad of things that you study when you study a person with an impairment is also the range of neuropsychological functions. So you pointed out you need to look at the other cognitive functions, so you need to do psych evaluation. We also want to know whether they hear properly. So if their information that they're getting in that they're working with is adequate and so on. So there's really, to answer a question like that, there's a range of data that you
Now that's another thing actually that's really important is hearing. And nobody asked me about this. Do they hear all right? And the answer is they do. And there's going to be a paper coming out which shows quite deep analysis now of this. So because, well, no, I'm getting ahead of myself. But anyway, look out for that because Farina's going to be writing that up soon. So it doesn't seem to be secondary to hearing problem. But you want to know this stuff, right? Could be a speech perception problem, yeah. I think, again, that's something they're looking into. It's a very interesting syndrome, I think. Yeah.
I think what we would really want to know, I mean, we would like to play our lives again without a speech problem and see if the more structural language problem arises or not. Yeah. So in order to determine whether it is or not secondary to the speech problem. Yeah. And this is, of course, impossible. Yeah. But we know many cases of children with speech problems who don't have linguistic impairments. Yeah. Even children who have congenital apraxia of speech who never speak once in their lives and who have perfectly normal language comprehension and language writing. Yeah, there are people with cerebral palsy who don't speak, but they're perfectly good at language. So we should not conclude from the speech problem that it is necessarily the cause of the linguistic impairment. Yeah.
What do we actually know about all the other speech patients? I mean,
no, but I haven't got to that yet, have I? So you're anticipating me a bit. But yes, we'll talk a bit about that, but much less. So we have other patients who are similar to the K family, but we know much less about them, is all I can say at this point. But I think we should. And one other thing that's really important here. This is the developmental phenotype. And these things are really difficult. They're especially difficult to study because it's a moving target. And mostly when we do genetic studies of kids with language disorders, sometimes we actually use quantitative scores in our studies. And people can shift, for example, from having a diagnosis of a specific language impairment when they're a kid to later in life it can become something more like dyslexia because they've, you know, and the scores can kind of shift around and the relationship between them. Interestingly, kids who start out with severe speech and language problems, often when they're younger, they keep up with their peers in terms of their non-verbal and verbal cognition scores. And something happens as they're getting older, and their cognition doesn't develop. This has been shown in several studies. So it might be that it's different. You know, there could be reasons for this impacting more broadly at different ages. So we need to just bear that in mind. So I want to that was an interesting discussion. I want to move on now to what we might go on.
One more thing.
Yeah.
I also think that we should not be misled by official diagnostic criteria.
Absolutely.
Because those diagnostic criteria have been designed primarily to exclude each other.
Yeah.
So like you define specific language impairment, but you exclude the speech impairment, which is nonsense because we know that in real life they are often
things go. Yeah.
But for some reason doctors are reluctant to make 2 diagnoses or 3 diagnoses for the same person, yeah. But I think they're wrong, at least those who are...
I agree with you. I mean, the idea that you can't have language impairment and speech apraxia is... Yes, and this is a quote from my friend Thomas Bourgeron, who always says that, and I think you know him as well, he says that the genes, they don't read the DSM, the diagnostic manual, so they don't know. So they don't know what they're supposed to do. Anyway, and let's move to genes. So we did a genome-wide scan in the linkage scan in the family using exactly this kind of techniques that I showed you already in terms of kind of color coding the chromosomes, but with gene markers. And we found that there was one section on chromosome 7 that was linked to the trait. So it was inherited by all the affected members and not inherited by all the unaffected members. And you can put a confidence, you can figure out how confident you can be in this with statistical analysis, because sometimes you might have chance occurrences. Because this is such a big family, we had the kind of LOD scores that we had were kind of through the roof, so very, very significant. And we mapped the gene to this. This is chromosome 7, and every chromosome has a kind of middle part called a centromere, and then 2 ends, telomere, 2 telomeres. And we localized the I've lost my. And we localized the gene to a section here. Now, this was in the days before genome sequences were available. This was, you know, late 1990s. And nowadays, you could just go to your computer. You probably do that now if you're on Google or whatever, and look up the sequence of the section of any kind of chromosome. You probably, if you go and look up 7q31, this is, if you go and type that into a genome browser, you'll pull out all the genes. You'll have them all listed, and it's great. We didn't have that luxury, so we were actually going through bit by bit, sequencing genes, and actually helping to build up the map that was eventually the sequence that was eventually called for this whole chromosome. And within this interval, there were a lot of genes. So something like 70 genes that were already known, and then a whole ton that weren't known. So how could we get past this? It's kind of tricky, right? We needed to zoom in quickly on one gene. Otherwise, we could be forever doing sequencing. Today, a company can just sequence the whole genome for you in a couple of days. But this was a long time ago. And we came across this case, and this is a child called CS, or coded CS, who gets much less attention in the literature than the K family, but he was very, very helpful for us to actually pinpoint the gene that was involved. And CS, the reason that CS came to our attention was that Jane Hurst, who was a clinician, who had been one of the first people to actually see the KE family. She took the blood samples from the KE family years before. She said, this kid just reminds me of the KE family. And as it happens, he has a translocation. Remember I talked about those big chromosomal rearrangements. He has a translocation on chromosome 7. And it's something that was diagnosed in pregnancy, and his rest of his family are normal, but he basically has the same kind of speech apraxia and a range of different language problems as the KE family does. And the curious thing was, so what led Jane to be interested in this kid was just that she thought wow, this phenotype is so similar. But as it happened, he had a translocation that was slap bang in the middle of the place where we were already looking. And this just shows you how you can, well, you can't see it so well, but just to tell you that this is how chromosomes actually look when you know, the earlier one it's kind of all neatly arranged, but this is what they look like under a microscope. These are all condensed because they're in something called metaphase. And this is chromosome 7. And let me get it right. Yeah, this is chromosome 7, right? So up to here, up to this yellow mark here, that's chromosome 7. And then after it, it's chromosome 5. And similarly, over here, that's chromosome 5, and then after it, it's chromosome 7. So there's been a reciprocal exchange of material. And this kid has all the genes that he needs. He has all the normal genes. But there's a breakage. Where does the breakage lie? On chromosome 7, 7q31, slap bang in the middle of this region. So our hypothesis then was that the reason that this guy looks similar to the K family is the K family have something going on in the same gene that is being disturbed by this translocation. So we had to actually characterize this gene. It was a new gene. We sequenced it, we put it all together, and then we sequenced it in the K family and looked for mutations. And we found that there was a single base change from a G to an A that was found in all the affected individuals, but not in the unaffected individuals. And the key thing here is these are old-fashioned DNA traces from DNA sequencing, and there are different colors to sort of show the different letters to help you see. But here, I don't know how well you can see it, but it doesn't really matter. The point is that there's two different nucleotides that are being read at the same time. And the reason is that one copy of this gene is normal, it's got a G, and at the same time the other copy is being read and you're seeing it overlaid and it's got an A. And that A, we then looked for in a ton of different people in the general population, and we never saw it. And we established that it's probably in the grandma in the pedigree. She's the first person to have been recorded who, I mean, we couldn't go further back because we didn't have any DNA samples, but she has it, and then she's passed it on to all the affected people in the next generation, and then they've passed it on to the affected people in the next generation. Okay, so that's nice. What is this gene? This is a gene called FOXP2. Now, it was a new gene at the time that we found it, but it belonged to a family. This is where evolution is very important. I didn't say that in the paper about it, but because if you find a new gene and it belongs to a member of a gene family through kind of historical events and things like that, then you can already say something about what that gene's likely to do. And what this gene is called FOXP2 because it has, in the protein that it makes, this is a picture of the, very simple picture of the protein that it encodes, 715 amino acids. Every protein has a start position, which is the N-terminus, and a C-terminus over here. And then you don't need to worry about these other bits, these other domains. This is the bit you really need to look at. This is called a fork head box domain. There are about, I think, 42 or something different fork head box genes in the genome of a human, and they all get different names based on the fact that they share this bit. They often, doesn't matter what's going on in the other parts of it, but this is the bit that joins the family of genes together and the family of proteins together. So what we could say already was that although it was a brand new gene, we could kind of line it up with other genes that were already known. And we also knew a little bit about what this domain does. So the domain in these fork head box proteins enables these proteins to stick to DNA. And I will get back to why that's important a little bit later. So this is a very crude schematic, but I like it because it's simple and conveys the idea of what this part of the protein looks like. And they're sometimes called winged helices. What you see here is 3 helices here, alpha helices here. And as I said, this thing likes to stick to DNA. And what happens is this part of the domain gets buried into the major groove of the DNA. And this binds to certain places in the genome because it's a regulatory protein. The K family have this G to A transition. Remember I said that we can say something, if something's hitting a protein coding gene, we can make predictions about what the consequence will be for the function of the protein. So this G to A transition is within this fork head box domain. it changes, it's a missense mutation, it changes an arginine residue at position 553 from the start of the protein to a histidine residue. When it does that, which is where it's shown in red here, it interferes with the shape of the helix, it interferes with the shape of the domain, it stops it from binding to its normal target. We've shown this in a ton of experiments now. So the KE family, they have 2 copies of this FOXP2 gene. The affected members have 1 damaged copy. It's making a protein that has this weird missense change. It then doesn't work like it should normally. We then went on and studied other families, and we found a few more cases, mostly isolated cases, but occasionally you can come across a family with a FOXP2 mutation, and this is the one we know most about here now, and it's called R328X, and the reason this mutation is called an R328X is it means that at a position 328 amino acids from the start of the protein, there is a stop signal. What that means is this is a nonsense mutation. This mutation leads to a protein that's severely truncated, missing a lot of important stuff, including the DNA binding domain. And what you'll notice here is, look at this tiny family, 2 kids affected with speech apraxia, and then a mum who has a history of speech and language problems, but we don't have a sort of detailed diagnosis on how she was when she was younger. But we know she had a history of problems. The father, who doesn't have the mutation, didn't have speech and language problems. Look at this family compared to that family. So had we just been going in here and wanting to find a mutation, it would have been quite tough. So we were very lucky to have access to a family like this. Most families look like that, actually, or sometimes just the odd case here. And there's quite a lot of work going on at the moment this is an Australian family, and we have some collaborators in Australia who are studying them. But it does seem like actually they have maybe more severe effects than the KE family. But they came to our attention because they had a diagnosis of speech apraxia. Yes?
Because the phenotype is so heterogeneous, and because what we know about complex genetic disorders like non-Mendelian, and also the complexity of brain development, if you look at the genome of the KE family, do you find other variants that are modulating the phenotype?
Well, that would argue then that. So what I think is interesting is that the KE phenotype within the family is for this type of disorder is reasonably consistent, actually. So I wouldn't want to. You can do a thing called you can do a kind of modulation screen. So you look for other genes that might modulate the severity of the phenotype. So if we had people in the family who had this R553H mutation, but were much severer than others, then it might make sense to do it. But I don't think we have enough power to look for modifying phenotypes. But the problem here is we have 2 things going on. One is that they have different types of mutation which could affect the severity of the phenotype. Right? No, if we're comparing across these. So, but the other thing is that they're on different genomic backgrounds, they're in different social situations, that kind of thing. But I guess, and I just want to say that we can study these mutations. We don't just assume that the mutations are functional, we study them in cell lines and other systems to try and establish whether they're really likely to be causing a disorder. This is really, really important for the future because as genome sequencing becomes cheaper and cheaper, people are finding more and more of these kind of mutations. You really want to know that it's causing a disorder. So here you can see if you take cells, you can grow human cells in the lab, you throw in different versions of your gene and you express them in a way that means you can visualize them under the microscope. So here's some cells with the nucleus stained in blue. And here you can see that FOXP2, normally FOXP2 likes to be in the nucleus, and the reason for that, it has special sequences that send it to the nucleus where all the DNA is, where this is the protein, the FOXP2 protein has to stick to DNA and regulate genes. So it needs to be in the nucleus. If you look at this mutation that interferes with how, in the KE family, with how the DNA binding domain works, you can, most of the time actually you see patterns like that, but sometimes the mutant protein can't get into the nucleus, and you see it in the cytoplasm. This version, the truncated version, never gets into the nucleus. It just sits around in the cytoplasm, and the reason for that is that it's lacking the special sequences that say put me in the nucleus. So this is not surprising, but we confirmed it in the lab. In addition to these point mutations, they're called, there's other, this is one that was published by Larry Schreiberg a couple of years ago. And there are others that are unpublished. They're all kind of, unfortunately, they're not in big families, and they're often not very well phenotyped. So we need to just do more and more studies on what the nature of the phenotype is in these other families and also in the KE family. And there's the CS case, and Larry Schreiberg also and Bruce Tomlin identified a family where a mother and a daughter both had a translocation that splits the gene in two. And again, they see the same kind of problems, speech apraxia plus linguistic problems as well. So I think we can be confident in saying 2 functioning copies of FOXP2 are needed to acquire proficient spoken language. And if you have damage to one copy, that is sufficient to cause a severe speech and language disorder. And we haven't yet found a case where, which is kind of, can reject this hypothesis, but we'll see what happens. So now I want to return to this, my favorite sort of slides. What I'm going to do for the next kind of 10 minutes is to show you how having found something interesting at the DNA and at the protein level, can we push a little bit to get into the middle parts? And what can we learn about FOXP2 and what maybe that can tell us about the disorder? So at the protein level, I've already told you that this is a regulatory gene, and it's a special type of regulatory gene called a transcription factor. And the way this works is, if we imagine this is, again, my very sophisticated view of DNA here. Remember, every gene has a promoter in front of it. Something comes along and switches the FOXP2 gene on in cells, and we'll talk about which cells in a minute. So it gets transcribed into messenger RNA, and remember I said all that stuff about it gets spliced and other stuff goes on. But ultimately, that leads to something called the FOXP2 protein being built. That FOXP2 protein which is built in the cytoplasm, then gets transported into the nucleus. It finds particular sites in the genome, and the sites that it finds, it binds to their regulatory regions. And then it might, for example, activate a downstream target gene X, or it might repress it, it might silence it. So this is we were talking about PAX6 earlier, this is exactly what PAX6 does as well. So there's a whole class of proteins in the genome, which are transcription factors, and there's lots and lots of different types of transcription factors, and they have different sets of targets that they like to regulate. So one thing you want to know is what are the targets that these things are regulating. Now, there's something here that, this is again an oversimplification in many ways, but we think that most of the time what FOXP2 does is actually repressing the genes that it binds to, and we actually think it doesn't act like it totally switches them off. You should think about it more like it's a dimmer switch. It's tuning the expression levels. And it's tuning the expression levels of many different genes. And what we've been doing for the past decade is pulling out using this as a kind of fishing hook to pull out the neurogenetic mechanisms around them. And we can do a lot of this stuff just with cells that we grow in the lab. And through that kind of work, here are just some of the examples of some downstream targets that FOXP2 is known to regulate. And I'm highlighting 4 examples here just because they're example genes which have also been implicated in neurodevelopment in other ways. And those are ones that we pay most attention to in our research program, just because we want to get entry points into the pathways. And when we don't know what genes, what downstream targets do, it's a bit harder to figure it out. You'll also see that I've got these A, B, C things here. So other things that we know less about but that we're trying to understand is what are the things that are upstream of FOXP2, what switches FOXP2 on and off, those might be interesting. What are the signaling pathways that might modulate how FOXP2 works? And no transcription factor ever acts by itself, it's always part of a complex of transcription factors. To understand that, you can see what are the proteins that actually bind to FOXP2 to help it work. What I want to point out with these genes, this is a gene called contactin-associated protein-like 2, which challenges my speech sequencing system. So CatNap2 we call it, and then there's a gene called MET here, and a gene called SRPX2 UPAR here, and a gene called UPAR here, and a gene called DISC1 that's famously been disrupted in schizophrenia. The point here is that these targets have all been implicated in a range of different neurodevelopmental disorders, And this brings us back to the point that I made and that Frank made as well, which is that genes don't read the DSM criteria. Many genes now we're finding bridge different neurodevelopmental phenotypes. And there's a whole discussion we could have about that, but we don't really have time for that today. And to give you another example of how this might happen, here is a gene called FOXP1, and maybe some of you, hopefully most of you, looked at Fabian's poster because he works on FOXP1. And this is the most similar gene in the genome to FOXP2. They stick to each other and they form what's called heterodimers, and they work together to regulate targets in certain places. This is an interesting thing to find because when FOXP1 was first found, there wasn't kind of a disorder to link it to. And we were studying the fact that it interacted with FOXP2, and eventually, at some point, a disorder came along, which is that it was found that in rare cases of autism and intellectual disability, there were these de novo, these new mutations popping up that just wipe out the function of FOXP1. And these kids have autism and intellectual disability, and most of them also have severe speech and language deficits within that. So it's a much more severe and wide-ranging disorder than the FOXP2 associated disorder. But it's interesting that there's some kind of functional connection there. Here's another example, a protein called TBR1, also a transcription factor, which also sticks to FOXP2. So what I'm trying to give you here is a flavour of how you can finding a gene that's implicated in a disorder is a starting point for getting into these kind of networks, and understanding the networks, and then also mapping those in relation to other phenotypes, other disorders. And there's lots I could have said about. There's a whole talk about CatNap2 that I don't have time to do today. But come and talk to me afterwards about it if you're interested. So what we see from this kind of protein level work that we do is that FOXP2 could be viewed as a kind of regulatory hub in networks that happen to have been implicated in a range of neurodevelopmental disorders, including things like SLI as well. Okay, so what about the cellular neuronal level? Here we can grow human neurons, or we can grow mouse neurons in the lab, or we can work with mice that have mutations and get neurons from them. And what we want to remember, I said near the beginning that there are different ways that genes could affect the neuronal properties. One of them might be proliferation, one of them might be outgrowth, you might think maybe plasticity, synaptic plasticity, those kind of things. So we want to have a readout at the neuronal level, or at the cellular level, that we can map back to the genetic level in order to kind of get close to the function in cells and in circuits. And what was curious was when we started, as I said, we started looking at these downstream targets. What are the targets that's FOXP2 switching on and off? And there's a lot of them, which makes it somewhat difficult to pull apart what's going on. But you can look at whether certain types of genes are overrepresented or underrepresented in those lists of targets. And what we found, we took cells from mouse brains, from a site of mouse brain tissue where FOXP2 is very highly expressed, which is in the precursors of the basal ganglia, right? So in striatal precursors, actually. So we were taking striatal tissue from mice. We were looking for genes that were regulated by FOXP2 in those cells. And we found that there was a big overrepresentation of genes that were involved in neurite outgrowth and in axon guidance. And these are genes that had been linked in prior work, in prior kind of mechanistic work. So what we wanted to do then was to culture these neurons, these striatal precursor neurons. And we had neurons where they have normal FOXP2, and then we have neurons where they have no functioning FOXP2 at all. So we've completely got rid of functioning FOXP2 in those cells. And what we wanted to ask was, could we see whether there were effects on neurite outgrowth in this system? Because that's the prediction from knowing what the target genes are. So we went from we used the the target gene analysis as a hypothesis generating tool, and then tested this hypothesis of neurite outgrowth. And what you can test with neurite outgrowth is like the number of these neurites that come out from the cells, And you can also test the amount of branching that they produce. This is work that was done by Sonia Vaness back when she was in my lab. And she now runs her own group working on bat genomics, amongst other things. What you can see here is that the normal cells have a normal number of processes, but they have much reduced branching compared to the mutated cells have much reduced branching compared to the normal cells. And this results in them having reduced process length. And this is really hitting the system with a big hammer, by the way. So this is not arguing that in the brains of the KE family that they have reduced neurite outgrowth, but it's a way of confirming that FOXP2 in this system might be regulating something to do with neurite outgrowth. You can read that paper if you're interested. This is just one of the ways of getting into the pathways that are involved. So a total lack of functional FOXP2 in precursors of striatal neurons leads to shorter neurites with reduced branching. Now, we actually also know what happens when you have a total lack of functional FOXP2 in an animal, because we've made mice. So we've made mice that carry the same mutation as in the KE family. They carry the exact same mutation. And there are also mice that have other types of dysfunction that knock out FOXP2 in different ways. And what we find is that if a mouse has no functioning FOXP2, it dies, and it dies after about 3 weeks of life. And those first 3 weeks of life, it's kind of got developmental delays. It's generally kind of severely affected. It doesn't vocalize much. I don't think that means a lot. It has a small cerebellum. So that tells you what happens when there's no FOXP2. And we've never seen a human being with no FOXP2, which maybe is not so surprising because it seems to be incompatible with life. All the humans that we've found have one damaged copy and one normal copy of FOXP2. All the humans with FOXP2 disorder. And when you look in a mouse, you can ask where, and you can ask this in a human as well, where is FOXP2 switched on, right? So remember I said that genes can't be switched, you can't have the same gene switched on in every single cell of the body, otherwise you get a kind of cacophony of gene expression. So to understand the function of a particular gene, you want to know which cells it's switched on in and which cells it's switched off in. And if you look at FOXP2, it's expressed in the brain, that's for sure, and I'm going to tell you a bit more about that in a second. It's also expressed in certain places elsewhere in the body. So it's expressed in a particular cellular layer in the lungs, particularly in early development. It's expressed in the gut, and it's expressed in the heart in certain places. And we might get back, I don't know if we'll have time for discussing it, but it's interesting to think about why is it that if you have no FOXP2, you die, presumably, maybe because of problems in hearts and lungs and things like that. We're still unclear about that. But if you have damage to one copy, what you see is normal heart and lung development, right? But effects on the brain, which are seem fairly specific. This is not actually a weird thing in developmental biology or in the fork-head box genes. There are other cases where there are genes that if you have one copy of them, it affects only one part of the body, but not others. Now, the reason for this is because certain tissues, certain cells are more dependent on the amount of this protein than others. So there's a kind of sensitivity. So what you're doing is you're halving the dosage, the amount of normal, healthy protein in these cells. And most cells seem to get by fine, but some cells in the brain don't like this. Which cells in the brain is FOXP2 expressed in? Let's see. So this shows you some early work that we did with Andy Kopp in the Institute of Child Health in London, looking at whereabouts FOXP2 is expressed in the brain. This is mouse sections, transverse sections from a mouse brain at 16.5 days of embryonic development, and I'm also going to show you the expression in newborns as well. It's not so clear to see, but I will talk you through it. So because the main take-home messages are the following. FOXP2 is not, some genes are expressed all over the brain. FOXP2 is not one of those. It's not expressed all over the brain, everywhere. However, it's not expressed just in one structure or in one spot. It's expressed in a number of different places in the brain, and where it's expressed, it shows some interesting specificity to particular types of neurons. So, for instance, in the cerebellum, this is not the best picture to show in this light, but anyway, in the cerebellum, it's mainly expressed in Purkinje cells. So the cerebellum is an extremely highly complicated structure with many, many different types of neurons and support cells all interacting. FOXP2 is highly expressed in a load of Purkinje cells, and this we see in mouse and we see it in humans, and we see it in all species that it's been studied in so far. It's expressed in dorsal thalamic nuclei. Some of the most interesting places where it's expressed is as I say, the developing striatum, in particular the caudate nucleus here you can see. And in the developing cortex, it's expressed in deeper layers of the cortex. So it's expressed in layer 6 mainly, a little bit in layer 5 and motor regions. And it's actually become a marker for deep layers of the cortex. Many people use FOXP2 just as a regular marker. And you could use it as a Purkinje cell marker as well if you want. And it's also expressed in the inferior olives in the medulla. There's a few other places as well, but what's interesting here, or what was interesting to us, was that it seemed to highlight corticostriatal and kind of olivocerebellar circuitry. And it was interesting to us that striatal and cerebellar circuitry was already known to be important for aspects of motor skill learning and sensory motor integration. And we thought this was interesting in light of the fact that the problems in the KE family, one of the most prominent problems in the KE family is speech motor sequencing and speech motor learning, if you like. And what I should also emphasize is this is really highly conserved expression pattern in vertebrate evolution. So in lots of different species, this kind of corticostriatal and corticocerebellar circuitry is showing up as being FOXP2, a site where FOXP2 is highly expressed. So this means that those are the sites which are potentially vulnerable to damage when the gene goes wrong. Now then you want to know a bit about what's happening to those circuits and how are they functioning in a living organism. Now you can't go around chopping up the brains of the KE family or sticking electrodes in. You can do MRI and MEG, which I'll maybe talk about a bit later. But you could do it with mice. And so we made, as I say, we made mice that carry exactly the same mutations in the KE family. And then and what we found was when a mouse has one FOXP2 copy damaged and one that's normal, like the KE family, it lives a long, healthy, happy life. You wouldn't really notice anything strange about these mice. They do pretty well on most behavioural tests that we've thrown at them. But we were particularly interested in motor skill learning. And we used a number of different tests, including a kind of classical rotor rod test, which I'll show you in a minute. But I like this test. This was developed by Matthias Grosser, who was in my lab, but now he has his own group in Paris. And this is great. This is a kind of tilted running wheel that you can stick in a mouse cage. And the nice thing here is that well, nice for me because you never need to pick up the mouse or force it to do anything. It just goes in the mouse cage, and it's connected to a computer so that every turn is recorded. So you get very, very nice motor patterning readout automatically. And it's a bit slippy, and it's tilted, as you can see. And mice, most people know mice love to run. So the lights go down in the animal house and mice run, or they jump on this and they run and then they slip off. And then they come back on and they run. So they run in a series of bouts. And the guys who originally developed this developed it because they were interested in cardiovascular phenotypes. So they use it for long-term training, like an in-cage gym for the mouse. And they monitor all sorts of stuff to do with how cardiovascular fitness changes over long periods. What we were interested in was the fact that the first time you put this thing in, and the mouse, naive mouse has never seen this, they have a kind of voluntary motor skill learning process that they undergo. And you can analyze multiple aspects of how their behavioral patterns change just in the first couple of days. And it's quite striking to see how they, so you can see firstly their baseline motor patterns, and then you can see how they how many bouts they run and how the bouts develop. And they end up running very fast and long bouts. In a fairly short period of time, they become quick. And what we found was that the mice with the same mutations in the K family were slower to learn to run on these things using this kind of technique. Now that's interesting, isn't it? Because that's not a orofacial test, although we might be looking into orofacial stuff as well. That's a kind of thing about general motor skill learning. And well, of course, there isn't a version of this so maybe think about what that might mean. Here's the data from this is work that was done by Rui Costa and Kath French, who are in Portugal. And what you can see here is when this shows you the fact that these are wild-type mice, and these KE mice are called R552H. It's exactly the same mutation as R553H in humans, just that the amino acid sequencing in the amino acid sequence in the mouse is just slightly one shorter. So there's a slight difference, but this is actually the equivalent mutation to the KE family. And what you can see is that this is an accelerating rotor rod phenotype. You stick the mouse on a wheel, and it falls off, and then you stick it back on, and then it falls off, and it's accelerating, it's high up, and they get better and better at doing it, and they go to sleep, and then you get sleep consolidation and so on. And what you can see is here's the KE mice, the mice with the same mutations in the KE family, and they never quite get as good as, and we've seen this now multiple times, multiple methods. But what Kath and Rui did that was nice that you could never do in the KE family was then to record from the striatum while these mice were doing going through these tasks. I'm not going to. If you're interested in this, then you can look at this paper here. But all I want to show you is that you can see neurons that are task-related neurons. So there are some that are positively task modulated. So these are medium spiny neurons that then are firing, increase their firing rate during the running on the wheel. And then there are negatively modulated ones that suppress their firing. And what you can see here are some recordings from the mice carrying the KE family change. And you can already see there's a few different things. I mean, dig into this paper because there's quite a lot of interesting stuff going on with these neurons. And there's definitely something different in there in the function of the medium spiny neurons in these mice. And you can see already at baseline they're overactive. On top of that, and we think this is an extra effect, there are differences in the modulation on the tasks. And we think this is not just a secondary consequence of higher activity, but something else that's going on. So they have the same number of neurons become task-related, but there are more neurons that behave in a kind of negative modulation way. As I say, dig into this paper if you're interested. And it shows you what you can do with one of these mouse models. And the idea here is that we try to link to a behavior. We're not trying to model speech and language deficits, right? We're trying to get into pathways that might be affected by this mutation. And this is a way to do it. And there's some other stuff that I haven't talked about, but the idea here is that you use this mouse model and it can give you some insights into this neural circuitry level that we can't do in the humans. And then we could probably take these findings back to the humans and ask what that might mean. And the other thing I should also say is that most recently what we've been able to do, we've been working on it for some years, is we have mice where we can inactivate FOXP2 just in the cortex, just in the striatum, and just in the Purkinje cells. And we've been kind of pulling apart the phenotype, and we have a paper that's not yet rejected. And so I'll tell you about that on another time. But we do want to be able to say something about what's going on in the brains of the KE family, for example. And there we see interesting convergence, I think. So what's curious is Kate and Fauner and people were doing brain imaging studies in the KE family when we had no clue what the gene was. So bear in mind, we didn't know anything about the identity of the gene. We didn't know where the gene was expressed. So in parallel, these kind of things came out. And what Fauner and Kate and colleagues found was when they did brain imaging on the KE family, they have, if you looked at their MRI, you wouldn't say there's anything particularly weird, right? But when you do voxel-based morphometry, looking at individual voxels in groups of affected KE family members against unaffected and unaffected controls, you start to see these statistically significant differences. And again, this is a very nice paper if you want to read it, but the key, perhaps the most interesting changes were seen in the caudate, particularly in the head of the caudate nucleus. And these are bilateral changes, reduced grey matter density, and also some slight increases in putamen. And there's effects on the left inferior frontal cortex and the left supplementary motor area. But the point here is, what I find curious about this is that the gene, we found the gene independently and we looked at where it's expressed. It's one of its most consistently highly expressed sites in the brain is in caudate nucleus and in medium spinal neurons. And it's interesting that this is a site that's that had come up in parallel in studies that without knowing what the gene was. Yes.
This is the fluctuation morphology.
Yes.
The fMRI.
Yes.
Then it's not so much.
Similar. So Frederick's study, which I'm not going to go into here, the fMRI study also, and also PET scans and things like that, are convergent in implicating striatal dysfunction and inferior frontal gyrus involvement. So I think that, but the question of what the connection is between those is really, really unknown. So the problem is, so for example, in the first study, the PET study showed overactivity of the caudate in word, I think it was word repetition task. And so, yeah, so there's a question of why should a region that's showing reduced grey matter density, I've got a feeling that there's something to do with the balance of caudate and putamen in development, but we're still investigating that kind of stuff. So I don't want to get too deep into that, but what I want to say is if we think about the structural phenotypes, we're seeing that these rare FOXP2 mutations, we've also done imaging on isolated cases. It's been very hard to publish those, interestingly, because they're case reports, and so they are in drawers, these studies at the moment. But we have other cases which seem to also implicate these same regions. But inferior frontal gyrus, caudate, and to some extent putamen, and also I didn't mention it on that last thing, but also there's some cerebellar sites come up as well in the structural data. This is the FOXP2 gene shown in a different way from before. And the reason I've had to change this is because I want to show you the introns, the gaps between the coding parts. So these lines are the gaps between the parts that code. Here's the DNA binding domain. Here's the R553H mutation. Here's the R328X8 mutation and the CS breakpoint. So what people wanted to know was, is there any evidence of effects in the general population? Remember I said that every gene kind of varies in everyone. Does FOXP2 vary? Well, it does, but not the coding sequence. The protein sequence is pretty much the same in everybody. It's very rare to find protein coding mutations. But there are variations. They all have these horrible names, RS22253. They're single nucleotide polymorphisms that are common in the population that we all share, and they lie in introns. And they are potentially the things that might be regulating how FOXP2 is expressed, but nobody really knows. But people have access to these sequences, so they go, yay, right, I'm going to get 10 people, stick them in a brain scanner, have a look at it, and see what happens when you carry these variants. And it's a legitimate hypothesis. Do common variants, we know that rare variation in FOXP2 influences brain structure, influences these speech and language skills. What about in the general population? So we, yes?
Do you see variants in the promoter matter regions?
Yeah, so these, sorry, I should say these are promoter regions of FOXP2. So this, I guess this might be considered, so there's a promoter here, so this might be considered a promoter polymorphisms, these two. But people really picked these out because they were available to genotype. And they wanted to ask this question. So what did they do? So what they did was quite a number of small studies and so the literature becomes a bit confusing because you're not sure. Nobody tries replicating anyone else's, and people point to different SNPs in different studies. So what we're trying to do in Nijmegen is go a bit beyond the small-scale studies to be able to do large-scale brain imaging genomics or cognomics, cognition genomics. And there's at least these people involved and many others as well. And what we want to do is have people where we have genomic data and we have structural scans, and we also, in some cases, and Peter's collecting a lot of this, we have functional data as well on interesting tasks. So what we have now is maybe 3,000 people with structural scans. About 1,500 of those, to nearly 2,000 now, have already had genome-wide genotyping, and we're going to do the rest of those in the next couple of months. So this is a resource where we can ask questions about individual polymorphisms, whether they relate to variation in brain structure. So what you see here is each of these SNPs that we studied, and the important thing, the only thing you want to read here is the number of people we did it in, because we're expecting these effects. You saw that the effects in the KE family were fairly, I guess, dramatic, but for common variation, you don't expect common variation to have massive effects, and also we're not expecting this to be causing speech and language disorders or global effects. We're expecting subtle effects. Prior neuroimaging genetic studies of these SNPs, various different ones, have studied up to 96 people. And we believe, and when you study small samples, we believe you're very susceptible to false positive findings. Maybe I don't know if Frank's going to talk about that later. And now we're getting to the point where people realize you really do need these large samples. And we started with the candidate SNPs and the regions of interest in the brain that were proposed in each of the prior studies. So specifically trying to replicate what was claimed in these earlier studies. We didn't replicate anything. And then we did just a gene-wide test where we looked at every variation in FOXP2 that we could find. And there we targeted the brain structures that were involved in the KE family in particular, so inferior frontal gyrus, striatum, and cerebellum. We didn't find anything significant. Now it could be that there are more subtle things that we haven't been able to detect in this sample size, but the take-home message is publish your negative results, because it came out in Frontiers in Human Neuroscience, and I think it's been a useful addition to the literature. Okay, so that's the end of this part. I'm going to spend a few more minutes on something else if I've got time, because I started a bit late. Have I got 5 minutes? Yeah? Okay, so rare mutations but not common variants of FOXP2 have effects on brain structure and function, overlapping with regions of gene expression. Just to get us going for this afternoon, I want to ask, you notice I haven't really mentioned evolution much. And some may think that means that I'm not so keen on evolution, but I am. So let's think about evolution. And what you've already hopefully realized, because I told you about some mouse studies, and I told you that it was really, that the gene was well conserved in mice. I also told you that the places where it's switched on are affected is the same in mice. And I also told you that striatal neurons are behaving weirdly in mice and also behaving weirdly in humans. So FOXP2 is ancient history. It's found in mice. I'm not going to show you this in the interest of time, but we've heard a lot about songbirds previous days. There's some very nice research by Constance Schaaf's lab where FOXP2 has been knocked down in area X because it shows dynamic regulation in area X of a zebrafinch brain. And you've already heard a lot about these kind of processes, and you know about HVC and so on. Fabian's work involves targeting HVC for FOXP1. But the idea here is that when people have studied FOXP2 knockdown in songbirds in area X, they see phenotype. I won't play you the sound files, but in the interest of time. But have a look at this Haisler et al. PLOS biology paper. It's really, really nice. So it seems to be important for some aspect of song learning neurobiology, even though we're very distantly related to songbirds. And there are people working on FOXP in flies even. So FOXP2 has been around a long time. In invertebrates, there's a single FOXP that has duplicated a few times since the evolution. But the curious thing is, even though there's only one FOXP that's related to FOXP1 and FOXP2 and FOXP4, it's doing interesting things in the brains of flies, and it might tell us something about the function of FOXP2. At least many of us hope that it will. And there have already been 3 papers on this. So we're talking about a deep homology, I guess, at some level, that FOXP2 is around, sitting there in circuits, ready to do interesting stuff. So whatever it's doing in humans is built on a very deep kind of history. But I feel like this is just obvious. I don't know, I was looking at this slide again and I was thinking, this is kind of obvious, but maybe it's not to everyone. But that we have a unique capacity, I think, but it has to be built on systems that are evolutionarily ancient. And that's what we see with FOXP2, is this is a gene that's been around a long time. However, even if you accept the fact that there's these kind of deeper history, right, there's still a question, right, which is that perhaps by comparing these two, given that we know that these guys are pretty bad at vocal learning, I think we agree on that, right? And we're very good. So at least at some level, we might say we should be able to do the comparisons of the human genome and the chimpanzee genome, and this might tell us something about recent evolutionary changes that have been important for tweaking the language system or for building up this language system, tweaking the components and bringing them together in a certain way. There's actually, we now know a lot about this, in fact. So if you want to ask a question about how does the human genome differ from the chimpanzee genome? So who knows, you remember we've got 86 billion neurons. I'm not going to do a big long calculation, but who knows how similar we are to a chimpanzee in the genome level? I'm sure everyone knows, right? No?
99%.
Yeah, it's a lot, right? It's pretty. So if you take here's a stretch of, I don't know, human or chimpanzee genome. Here's a stretch of the other genome. We can line those sections up. We can identify the direct equivalents, and then we look and we see, look, there's a difference. About 1 in 50 there, something like that. Zoom out a bit. There's 3 changes in about 200. We're at 98.8% identical to chimpanzee. That's a lot, right? Or is it a lot? Because the genome has 3 billion letters, 3,000 million letters. If you count it up, that's 35 million substitutions. That's a lot of room for stuff to happen, right? We don't know what a lot of this stuff is doing. But another point is that actually, this is only for the bits we're able to line up. There are plenty of bits that we can't line up. What do we do when we have bits that we can't line up? Well, we just kind of stick them in and where they are and kind of go, okay, there's been an insertion deletion event. If you do that, we're 95% identical. Now, a lot of that stuff's very hard to study, but so more than 5 million of these events happening through the genome. And lots of changes in genomic regions of unknown function. So the point here is that you can play this whichever way. You can say, oh my God, we're so similar to chimpanzees, nothing genetic could ever be of any interest. Or you could go, wow, look at all this stuff. And the other thing that's interesting is, even in the initial sequence of the chimpanzee genome, you want to know what makes us human. It's all there, right? If you want to know genomically what makes us human, we have a complete catalogue of all the genetic differences that have accumulated since the human and chimpanzee species diverged. As I said, it's a lot, 35 million single nuclear changes, 5 million insertion deletion events, various chromosomal rearrangements. The problem is, we have it all, we just have to figure out which bits are important and what and how they've changed and what they relate to. Now, one way we could, yes?
I've got a question. So 95% is still a lot, even.
Okay, well, but
yeah. And you say there are loads of rearrangements and so on, but rearrangements also occur within humans.
Yes, oh yes.
So
yes, that's, yeah.
So how different
yeah.
would two humans
be? Two humans are a lot less different. Okay. That's true. And actually that raises a general point, which is what we were doing there was comparing a sort of template of the reference genome for a human, which is now much, even in the past 10 years, it's changed a bit because we have that, and that's being compared to the reference sequence for a chimpanzee, which is based on a very small number of chimpanzees. It's beginning to be expanded with more, and now we have, as I told you earlier, we have a lot of ideas about the ranges of variation. So we can start to ask questions about the range of variation in humans, the range of variation in chimpanzees, how many changes are fixed. So the most interesting things are the changes that are fixed and don't vary between humans. But there's much less diversity between two humans than, dramatically less than so again, that's how you want to play it. But these kind of figures are kind of, they're not very helpful, I think, because I'm not sure if this is exactly right, but does anyone know how similar we are to a banana? If you do. Yeah, something like 50%. But I've been trying to track down where, because I Googled it, but I haven't been able to figure out who's actually who's done the proper comparisons. But we are very high, if you do this kind of analysis, it doesn't mean a lot. It's really about the natures of the changes and where they are and what they do. Yes?
Just to complement to add to this story, if you think the genome is very, very similar, there's this idea that what actually will drive evolution is change in the gene expression level. So the phenotypic, the genomic differences are, there are not that many genomic differences, but if you look at how this gene is expressed, actually there could be a lot of change there that is driving the phenotypic differences.
But that's where the changes are then, you see. There have to be changes in regulatories. But anyway, because I'm running, I'm going to come to the end, but I also want to say that we also have, for those of you who are interested in hypotheses about recent evolution of language, we also have, this goes back to the point Dan made, we also have a genome sequences for Neanderthals and for Denisovans. We never thought we would have, we didn't even know about Denisovans until very recently. But there are other ancient hominins that we're this twig that's left from a very bushy branch, and maybe we start to look at some of the other twigs on the branches that we can access, which is very tricky to do. But we do have genomes of Neanderthals. And if you think about it, it's like a few 100,000 years ago when we diverged from Neanderthals. And I want you to just look at what, this is work by Svante Pääbo, and there's more and more coming out all the time. We have a definitive list of substitutions that became fixed in modern humans after the separation from the ancestors of Neanderthals and Denisovans. And what does this look like? So this is, this is kind of a phylogeny that's currently thought for modern humans. Neanderthals branched off here, and Denisovans branched off from this strand here. And the idea here is that we have, if you want to know what's changed within the past 100,000 years, or the past few 100,000 years, we know them. We have this list of 31,000 single nucleotide substitutions, 4,000 indels. These are small indels, by the way, not the big ones. 96 fixed amino acid changes, 3,000 changes that potentially influence gene expression. So we have this list. So if there is a gene that was responsible for, if you want to find the merge gene, it's here. It's there. And this is, so I think people should put their money where their mouth is. And I think that if people who make strong claims about a single major event that created language or created some aspect of language, then it's in these data, and I think it can be proved. And I really like the fact that we're headed towards an empirical science for language evolution. And I'm just going to finish by saying that even though FOXP2, we heard a bit about this already, even though FOXP2 is very, very well conserved, it's been around for a long time, there are 2 amino acid changes that occurred to the FOXP2 protein after we diverged from chimps. Against a general background here of not much stuff going on. There is a change in orangutans, which maybe somebody should study, I don't know. And this, I'm gonna just, there's also changes, as well as these amino acid changes, there's regulatory changes that have occurred after we've split from Neanderthals. So though Neanderthals shared these amino acid changes, there's been subsequent changes that have happened in regulatory regions. And I won't talk any more about that, but just to say that this is the way we should be approaching the evolutionary thing, is not in a kind of naive, we should approach it from, it's not either that a gene's been around for a long time, or that it's super special. It's that genes that have been around for a long time can show interesting patterns of change in relation to human evolution, and we should try and understand that. Okay, so I'm going to just end by saying heterozygous mutations in FOXP2 cause severe speech and language disorder. It's a regulatory gene, its downstream targets and interactors give entry points into neural pathways involved in speech and language, and it's not a gene for speech, in my opinion, because we find it or a gene for language, we find it in many vertebrate species doing interesting things, so that's how we should always frame these kind of discussions. And we have particularly lots of clues into what it might be doing in corticobasal ganglion circuitry. We've still got masses to learn about its functions. And I will stop with this slide, which has quite a few of my collaborators, but it's been a very, very big effort involving many, many people. So I'll thank you, and I'll thank you for your attention.